Welcome once more. Do not forget that you are in the Greens Academy in lesson 4. In lesson 4, we shall look at some important aspects of enzyme action. We are just from talking about the properties of enzymes in lesson 3. That will help you to understand lesson 4 very well. So, lesson 4 gives you a real picture of enzyme action, showing us the different facets that are involved in enzyme action. Then, for an enzyme to work, a little amount of energy must be put to get the reaction started. We all know that this energy is called the activation energy. And we should not forget that enzymes work at reducing the activation energy required for a chemical reaction to occur. I equally wish to remind all of you before we get into lesson 4 proper that living cells carry out many biochemical reactions. These reactions take place rapidly due to the presence of enzymes. All enzymes consist of globular proteins which have the ability to drive biochemical reactions. Example, the enzyme dehydrogenase needs a coenzyme, NAD, to function. And we all know that NAD is nicotinamide adenosine diphosphate. Nicotinamide adenosine diphosphate. That is a coenzyme, because we should know that some enzymes need these coenzymes in order to work best. So, lesson 4 now takes us to the following heading: mechanism of enzyme action. Mechanism of enzyme action. Therefore, we want to see the ways or the manner in which enzymes carry out their actions. There are two main hypotheses for enzyme action. There are two main hypotheses for enzyme action. We have the lock and key hypothesis and the induced fit hypothesis. So these are the two hypotheses that explain fully the mechanism of enzyme action. We will start with the first one, which is the lock and key hypothesis. I guess you understand what we're talking about, because we have earlier talked about the lock and key when we're trying to talk about the active site and exactly what happens at the active site of an enzyme. We said that when you have a lock, you need a key to open that lock or to close that lock. And each key is specific to a particular lock. So now we are bringing in the concept of the lock and key hypothesis here for you to understand exactly how enzymes will function in this particular rubric. The key is considered as the substrate and the enzyme the lock. I will not emphasize on this any longer because we have had to explain this in previous lessons very well. You now know why the key is considered as the substrate and the enzyme the lock, because the key is inserted in the hole found on the lock to open it or close it. Lock and key hypothesis. Enzymes have particular shape into which the substrate fits exactly. This is important. Enzymes have particular shapes into which the substrate fits exactly. We have explained this in the previous lessons, because the shape of the active site should or must be in conformity with the shape of the substrate, without which the substrate will not be able to fit adequately in the active site of an enzyme. That is why any change in the enzyme's shape, I insist, any change in the enzyme's shape, no matter how small, changes its effectiveness. Changes its effectiveness. This is important. Here we know that the site of the enzyme has a shape that is ready to accept a specific substrate. That shape, we know that is made up of a sequence of amino acids. In the course of any change, if any change occurs at this site, no matter how small that change is, this will go a long way to affect the effectiveness of this enzyme, so that at some point in time, the substrate may not be able to fit again in that particular active site. The active site of the enzyme is structurally similar to that of the substrate. This is important. Structurally similar to that of the substrate. That's what I've been saying since, that the active site is structured such that it is in conformity to that of the substrate, without which they will not bind, without which no reaction will occur. So it must be similar in structure and shape. Both must be similar. That is the active site of the enzyme and the substrate structure. The substrate binds to the active site. The substrate binds to the active site of the enzyme. When this happens, a temporary structure called the enzyme substrate complex is formed. We have explained this, that when the substrate binds to the active site of the enzyme, a structure called the enzyme substrate complex is formed. And this is temporal. This is very short-lived, because in the long run, it will have now to lead to the substrate product complex that will now release the product and allow the enzyme free to carry out or catalyze other reactions. The products formed have different shape from the substrate, still under the lock and key hypothesis, because the product formed has a different shape to that of the substrate. And once these products are formed, they are released from the active site. That is why I said that when the enzyme substrate complex is formed, this now leads to the enzyme product complex. And immediately the enzyme product complex is formed, it is short-lived. It is very, very temporal. Therefore, at this point now, the substrate must dissociate. In other words, the product, I mean to say the product dissociates itself from the enzyme. This leaves the active site free so that it can attach itself again to another substrate. That is, immediately the substrate product complex is formed, the product now leaves the active site of the enzyme, thereby allowing the enzyme with its free site to now bind to another substrate to carry out another reaction. The enzyme and its active site remain unchanged. The enzyme and its active site remain unchanged. Remember, this takes us back to the definition of enzymes. Biological catalysts speed up chemical reactions and remain unchanged at the end of the reaction. You have to understand this now, because remember I told you at the beginning that the definition of enzyme is going to be involved in almost all aspects of this lesson. And you now understand why, after everything has taken place in the lock and key hypothesis, at the end, the enzyme and its active site remain unchanged. Now, looking at this diagram, which now explains fully the concept of the lock and key hypothesis, you will now see that substrate C binds to the active site labeled B, because the enzyme is labeled A. That's for the first diagram. Looking at the second structure, you realize that the enzyme A, which is in red, the substrate C, which is yellow, binds into the active site labeled B. And when this happens, you have now structure D, which is an enzyme substrate complex. When this has occurred, this will now lead to an enzyme product complex. And finally, since it is short-lived, it now takes us to E, where you now realize that the product dissociates or leaves the active site, making the active site free. And we said a few minutes ago that at the end of this reaction of the lock and key hypothesis, the shape of the product is different to the active site and even to the substrate at the beginning. So look at the two products here labeled F. Look at the two products labeled F. You will now see that their shapes are different to that of the active site and also different to the original substrate. And this gives us the concept of the lock and key hypothesis. And this lock and key hypothesis was suggested by Fisher in 1890. This hypothesis was suggested by Fisher in 1890. Now, we now move to the second aspect of mechanism of enzyme action. This second aspect is what we call the induced fit hypothesis. The induced fit hypothesis. It is good for us to understand this very well. Induced. The word induced should ring something in your brain. It means that something changes in the course of the reaction to cause the substrate to fit adequately into a particular site. Therefore, in this case of the induced fit hypothesis, the active site changes its shape to bind to the substrate molecule. The active site changes its shape to bind to the substrate molecule. What does it mean? This means that at the beginning of this reaction of the induced fit hypothesis, the shape, in other words, the structure of the substrate is not similar to the structure of the active site. Therefore, at the beginning of this reaction, the substrate cannot conveniently fit in the active site of the enzyme because they have different shapes. Therefore, for the reaction to occur, the active site must change its shape to fit or to look similar to that of the substrate. The following events are involved in this hypothesis. These are the things that occur in the course of the induced fit hypothesis. The first thing is that the active site has a shape that does not conform to that of the substrate. That is, the active site has a structure that is not similar to the structure of the substrate. Now, as the substrate approaches or moves towards the active site, the site now changes to the shape and structure of the substrate. This is important. I want you to understand this very well. At the beginning, the shape of the substrate is not similar to that of the active site. As the substrate is approaching the active site of the enzyme, the structure or the shape of the active site now changes to now match to that of the substrate. This results in a perfect fit. This will now result in a perfect fit because as the substrate was approaching the active site of the enzyme now had to change its shape and structure to look similar to that of the substrate so that a perfect fit can occur, so that it can bind exactly to that active site. After this reaction has taken place, an enzyme substrate complex is formed, which is short-lived, and this will lead to an enzyme product complex, which is also short-lived, and at the end, the product is formed that leaves the active site. And when the product leaves the active site at the end of this reaction, that is the induced fit hypothesis, the active site now returns to its normal original shape this is important, my dear students. This is important. Remember that at the beginning of this induced feed hypothesis, the enzyme has an active site which hadn't the same shape to that of the substrate. The substrate approached it and it now changed its structure to suit that of the substrate. Now, this was just to enable that reaction to occur. The induced was just to enable the reaction to occur. That is why at the end of this reaction, the enzyme now regains its normal original shape. You will now see this in the diagrams that follow. Now, when you look at this diagram, my dear students, I want you to observe the first diagram. There are 4 of them. Look at the first diagram. Look at the shape of the substrate, my dear students. Look at the active site of the enzyme. You now see that the active sites are not really similar in structure to that of the substrate. You now see that as the substrate is approaching the active site now change to suit the substrate. And that is why in the second diagram, you now realize that the substrate is well fitted now in the active site now that change to form the enzyme substrate complex. And when the enzyme substrate complex is formed, since it is short-lived, it now led to the formation of the enzyme product complex. This now is also short-lived and now had now to lead to the 4th diagram, which you see there, where the products leave the active site of the enzyme. And you look now critically that the products that are produced are different to the shape of the active site and are also different now to the original substrate that we had at the beginning of this reaction. And you look again that, you will now see again that the enzyme fully regains its normal shape at the beginning, its original shape. Because if you look at the second diagram of this structure, you now see that the active site now changed to accept the substrate. So that side now is different from the first diagram and the last diagram. So at the end now, the enzyme now takes its original shape so that it can go now and start another reaction or start another process or start another biochemical process. So this is where we shall end this particular lesson 4. And this now prepares us for the next lesson. Thanks for your kind attention.